Well, hi and welcome everyone to Ecology Live, our new series of online talks from the British Ecological Society. I'm Jonathan Wood, I'm Head of Society Programmes at the British Ecological Society. And if you're not aware of the BES, our society, we're the biggest scientific society for ecologists in Europe, with a membership of over 6,000 all around the world. And if you'll just allow me, my slides aren't advancing, so excuse me while I sort that out. Let me try again. So yes, there we are. Apologies for that. So yes, if you're not aware of us, we are the largest scientific society for ecologists in Europe. And we're broadcasting these free talks, of course, during the coronavirus lockdown period, so that there's still a great opportunity to hear the latest research from top speakers like the one we have today. And even while we're all working from home, we're approaching 3,000 registrations from people to this series, Ecology Live. And I can see well into the many hundreds of attendees joining us right now. So thank you everyone for that. I'll shortly hand over to our speaker, Tom Crowther from ETH Zurich in Switzerland, who's giving today's seminar. But just let me first quickly explain. There will be a short question and answer session at the end of the 25-minute talk. You can submit your questions during the talk. There's no need to wait to the end. And that you can do using the Q&A box. You can make your questions named with your name or anonymous, you can select. And just to note as well, we are recording the talk and we'll post the video to YouTube afterwards. So without waiting any longer, let me just introduce today's speaker, Tom Crowther, who will take us from fungi to biogeochemistry on the global scale. Thanks very much, Tom.
Hi, thank you so much for the introduction. Just going to share my screen. Yes, thank you so much for the introduction. I really appreciate, what the BS is doing is so fantastic, giving us all an opportunity to connect during this difficult time. And I really, thank you so much for inviting me to speak. I've been presenting a lot of late about our lab works on various different aspects of ecosystem functioning and how ecosystems both respond and then feed back to climate change. And much of my recent presentations have been focusing on the topic of trees and how they capture carbon from the atmosphere. But I thought this was a nice opportunity to talk about something that I've been working on for a lot longer time, actually, and that's these trade-offs in fungal functional traits and how they can help us to predict wood decomposition across broad spatial scales. And one of the reasons this is such a nice opportunity to talk about this topic is because this research started about 8 years ago in my first postdoc, working with Mark Bradford in Yale. And this all was initiated by a grant from the British Ecological Society, a starting grant from the British Ecological Society, which really kicked off this research. So it's, and we've had some recent developments in this field and this topic, which I can talk about today. So thanks so much to the BES. So I normally start my tree talks with this incredible NASA simulation, which shows beautifully how at the beginning of the year, when leaves emerge on trees in spring, they draw down so much carbon from the atmosphere and you see these concentrations fall. But it's equally important to show what happens later in the year, in autumn. As we tick on into sort of the end of summer and then autumn, we'll see the concentration starting to rise again. And that's because the leaves obviously fall off the trees in the northern hemisphere. And we see a lot of decomposition activity and the respiration of soil organisms and trees and plants across terrestrial ecosystems really bumps up atmospheric carbon concentrations in this natural cycle that we've had for many, many centuries. And of course, the scale of this is incredibly impressive. But what a concerning thing is that we think we're seeing higher losses of carbon into the atmosphere year on, year out, and that might be indicative of some biological feedbacks to climate change. And so ultimately, this is what we're interested in. It's the mechanisms or the storage of carbon over the rest of the century on land. And so many of the Earth system models, this is the classic paper by Friedlingstein showing how divergent our model predictions are when predicting future carbon storage. So many models expect that carbon storage is going to go up as a result of increased plant growth and productivity in a warmed environment with elevated CO2. But of course, many other models actually predict that carbon stocks are going to go down as a result of maybe increased tree mortality and plant mortality, but also because of losses of carbon from the soil under a warming climate, as we see elevated rates of soil respiration. And my own personal opinion is, in fact, that we might be overly optimistic in some of these models in that, first, I think the plant response might not be as strong as we initially thought it might be. And I actually have growing concerns that the soil response might be, in fact, larger than we expected. And so ultimately, that's very concerning, because when we think about the carbon pools on land, the soil is, in the biosphere, our biggest store of organic material. This graph shows the patterns of carbon storage in above ground and below ground, from the South Pole all the way up to the North Pole. And what we see is, obviously, there's a lot of carbon in the below ground world, but almost just as importantly, we see that there's a huge bomb of carbon in those high latitude regions, where cold conditions have slowed decomposition rates and respiration rates of soil microorganisms. And that has led to the accumulation of huge carbon stocks in those high latitude regions. And now the real concern is that as we warm those cold regions, we're going to remove the temperature limitations, which are going to explode, lead to a loss of carbon from the soil up into the atmosphere. I know many people know about this in permafrost regions, but of course, these northern latitudes extend far below, the UK, for example, where we've got very rich, carbon-rich soils. So it's a real concern. And we've been working on this topic for a long time with a range of different laboratory and field studies. But one of the most valuable breakthroughs came from a large data collection where we collected soil samples from many warming studies across the globe. And that allowed us to essentially quantify carbon stocks and carbon estimates in all of those warming sites. So unfortunately, we weren't able to get studies in the Southern Hemisphere, but we were able to collect about 49 studies where there's a long-term ecosystem warming experiment in the Northern Hemisphere across a range of environments. And these range from in various different ecosystem types with various different plant functional groups, but all of them have warmed plots adjacent to ambient plots. And that means for each of these 49 locations, we can calculate a difference. And that's the impact of warming on soil carbon. And we found that there was huge variation. In some places, there was increases in carbon under warming, and other places there was decreases. And what we wanted to do is try to explore the environmental drives of this to see if we can figure out which environments are seeing increases and which environments are seeing decreases. And so we correlated these effect sizes with all sorts of things like temperature and moisture and latitude and altitude and many environmental variables. And we were astonished to find that only one predictor came out as by far the strongest predictor. And that was actually the size of the standing carbon stock in the first place. Now, obviously, this is indicative of many other environmental characteristics, like the amount of moisture and temperature in the region. But what we clearly saw is that in the regions with large standing carbon stocks, as in a lot of carbon in the soil, those were the areas that are most sensitive, and those are the places that we're seeing consistent decreases in carbon stocks. But as we move up into the areas where there was smaller standing carbon stocks, we started to see negligible changes or even increases in carbon. And because we knew we've got a reasonable idea about the amount of carbon storage in each of the locations across the globe, we can extrapolate this very simple linear relationship to every single pixel around the planet to estimate where the vulnerabilities in carbon and soil carbon is to warming and which are the most sensitive regions. And there's where we see that those high latitude regions, the cold, temperature limited regions, are possibly the regions that are most vulnerable to changes in carbon stocks, as we might have expected. And by adding up all of the values in all of these pixels, we can get an estimate of how much carbon might be lost at any point in time. And we estimated that with a business as usual climate change scenario, that we'd be losing about 1.5 gigatons of extra carbon per year, which is a considerable addition to what humans are emitting right now, about 12 or 15% increase. Now, there's been a lot of research on top of this of late, introducing a huge amount of uncertainty and variability in this topic. But what it's important to remember is that this is simply the effect of warming. It's taken in isolation, not including the impact of CO2 and moisture availability that are also changing under global change. So what we really need to do is put those temperature sensitivities back into the Earth system models to really evaluate their impact in the context of other global change drivers. And I work with Kathy Todd Brown here, who's an exceptional mathematician and modeler, who essentially got those estimates for each of the Earth system models about how much the land carbon change by the end of the century. And this very simplified graph shows each dot represents one of those models and the projected amount of carbon on land, or the change in carbon on land by the end of the century. So a point above the line is an increase, and a point below the line is a decrease. And now what she figured out is how to essentially take our unified temperature sensitivity data and plug it back into each of these models. And what we see is immediately the carbon stocks fell. So the projections about the potential carbon storage declined. But almost more importantly, we actually see that there was a narrowing of the uncertainty, as now all of these models have the same parameter for this temperature sensitivity across the globe. However, we also now have uncertainty. As I mentioned, there was a lot of uncertainty around those graphs, a lot of standard deviation and error. And that means when we propagate that error, what we actually found is that we can propagate that uncertainty into every single model. And we find, in fact, that the variation within any single model can actually exceed the variation across all of these models, which highlighted to us that the variable uncertainty within these models can be equally, if not more important than the variability across these models. And that is something that we can really only constrain, I believe, with improved mechanisms into how these ecological feedbacks are taking place. So we need to better constrain those predictions. And from my own perspective, I think the best way to do that is to actually insert the ecology. I'm talking about these ecological feedbacks to climate change, and yet I've not at any point mentioned an ecological process. And so in order to do that, it's very challenging, of course. And I think what we can do is learn from some of the incredible advances that's happened in the above ground world in the recent decades. And I think two of the most important advances in terms of inserting that ecological information is first to break up the world into plant functional types. Now we can say at any pixel, if it's a light green pixel, that might mean it's a grassland, and a dark green pixel means it's dominated by forest. And that means you'd have very different carbon turnover rates in each of those pixels. And that's been an incredibly important step for constraining that uncertainty. But the other equally, if not even more important step is where we're at now, where we're trying to introduce variation in traits across across and within those plant functional types. We all know how even within a single species, you can get huge variation in plant traits. And imagine that within a functional type, you get huge variability. And that means by propagating that variability, we can start to say, right, this pixel, not only is this plant functional type, but it's got these characteristics. And that means the model can run in an appropriate way to represent those mechanisms. And so that's what we've been trying to start to do for the below ground world. And I think Brian Stedinger had a fantastic effort to sort of initiate the first of those functional group maps for us. Essentially, the breaking the world up into functional groups. And he started with the dominant symbiotic microorganisms with plants, showing the relative, essentially characterizing ecosystems based on their dominance of ectomycorrhizal or arbuscular mycorrhizal fungi, which have such different impacts on plant growth, nutrient availability, and carbon turnover. And now that charge is being led by Gabriel Smith in our lab, who's essentially put out a recent call for collaboration. This is a new kind of paper that I became aware of very recently. A call for collaboration. It's been fantastic. We've had such an incredible amount of people wanting to come in and collaborate and share quantitative data on these major functional groups of microorganisms that are happening in the soil. And so when we get somewhere with that, we're starting to build these maps that will hopefully be able to break the world up into these pixels. And with each of the pixels, we can run a slightly different equation to represent the relative dominance of those 2 or 3 or 4 or 5 major microbial groups. And that will have distinct consequences for how carbon turnover happens there. But where we're currently lacking, and where I want to focus on today, is in fact not the breaking the world up into functional groups, but the second of those things they've done above ground, to explore the patterns in trait variation. And this is an incredibly difficult thing for microorganisms, particularly at a large spatial scale, because as we all know, they are inherently tiny, complicated, and difficult to see, but they're also incredibly variable. And for all of these reasons, it's incredibly difficult simply to measure individual traits on an individual organism directly. We get proxies of trait expression at the community scale, and that's fine. like enzyme activity or respiration rates, but to get individual trait expression is very, very challenging. And so efforts that have done so, first, we struggle to even identify which of the traits we should focus on. You know, with plants, we've got the whole economic spectrum of plant form and function, which points us in the right direction. But for microbes, there's so many traits that we could focus on. And because of all of this complexity, it's led many people to believe that there aren't actually good predictive patterns or unifying patterns in the patterns of microbial traits below ground. And in fact, that they might be more stochastic or governed by neutral dynamics to a greater extent than environmental filtering. But to overcome many of these challenges, we've been working with a charismatic group of microorganisms, and that is the saprotrophic fungi, which dominate wood decomposition in forests and many other ecosystems as well. And they're important for the carbon and nutrient turnover in these ecosystems. But they're also charismatic organisms, and we've all seen their mushrooms above ground. But the real key to these organisms is the mycelial systems that happen below ground, where they are responsible for distributing nutrients across their ecosystems, for giving soil stability in its very structure that is important for other organisms, including forming aggregates, which are microcytes for microbial activity in the soil. And even the decomposition of this necromass, when the fungi die, that is one of the mechanisms through which organic matter stable soil organic matter is ultimately formed. And so these organisms and their mycelia are so important for the functioning of these ecosystems. And yet we would be very hard pushed to tell you what these mycelial systems look like in a very dry grassland or desert compared to a very wet forest. We have absolutely limited insights into the trait variation across our ecosystems and how these mycelial systems function. There are people that might say that, in fact, we tend to see more melanin and lignin in stressful dry conditions, but ultimately getting individual level trait variation has been very, very challenging. So we've been working with a large data set, well, in fact, not a very large data set. We'd like it to be a lot larger, but we want to measure so many traits on these fungi that we've had to limit the number of fungi we work with. But a data set of about 40 fungi from across North America that were collected from a wide environmental range. And these are a range of different species and from a range of different environments, but they were all saprotrophic basidiomycete or ascomycete fungi that were collected in stages 2 to 3 of wood decomposition. And they were either isolated from the decomposing log or from the fruit body itself. And that means that these are functionally equivalent dominant microorganisms. These are all very common in fungi in forests across this region. And they all would be likely to interact if they were in the same space, because they're sort of competitively equivalent. And they come from a very wide phylogenetic range, and having that phylogenetic structure is very useful for our analyses. But ultimately, we wanted to start to see if we could tell which types of mycelial systems live in which types of regions. And again, the very difficult challenge is figuring out where do we even begin. There are so many traits that we could start with. But we thought we'd go back to the very drawing board. And my friend Dan Maynard and I actually started this work, as I say, about 8 years ago, essentially taking each of these fungi and growing them across gradients of temperature, because we thought the simplest trait might be the niche space, at least the temperature niche space. We thought the individuals from warm conditions might have an optimum up around here in the higher temperatures around 30 or even higher, whereas individuals from the cold Arctic regions might have a lower temperature sensitivity or temperature optimum. And after a lot of plating and a lot of playing with these fungi, we came to realize how very little we knew about organismal physiology. Of course, almost all enzymes have an activation energy that increases up to about 25 or 35 or even higher, after which point they denature. And that is, of course, no different for these microorganisms. So in fact, they all preferred to exist in the nice warm conditions. So we thought, right, temperature is a bad environmental filter. Maybe they're underground and they're protected from changes in temperature, so they don't care. Drought, we thought moisture must be a much more valuable, much more meaningful selective pressure. And based on the literature, we really thought that might be the case. So we grew them across gradients of moisture and found exactly the same thing, that in fact, they all preferred to exist in nice moist conditions rather than the dry conditions. But there was one meaningful axis of variation that we found in our despair. And it's not the optimum growth point, but it's in fact the niche width. Some of the fungi had a very narrow niche width, which means they can only exist under optimal conditions. whereas others had a very wide niche width. And that means they can exist across a range of environmental conditions. And this sets up the framework for a classic sort of CSR or dominance tolerance type framework, where the individuals that can only exist under optimal conditions, they must be competitive and they must have to fight for those optimal conditions. Whereas the individuals that can tolerate harsh conditions, they probably can't be bothered investing so much energy in fighting, and they might just tolerate those harsher conditions. And that might be something that filters them across gradients. So we were very excited by this possibility, but also terrified by the fact that in order to test this, we needed to get a fungal hierarchy, a competitive hierarchy. And that ultimately means fighting every single fungus against every other fungus. And it ultimately meant many, many weeks sitting in front of our Petri dishes, plating in a little production line to initiate these little fungal battles where we had individual fungi fighting against one another so we could see which ones are dominant. And so we measured how quickly they grew towards one another. We measured how quickly they grew away from each other, or even over one another, or underneath one another. And we came up with various metrics for how competitive they were. But we ultimately were able to collapse this. And I can answer questions forever about how we characterize that hierarchy, because it took us a very long time. But ultimately, we came to what we thought was our best version of this competitive hierarchy. And after a long, long time of plating, we were delighted to find that we finally did find evidence for something meaningful in our data set, which was evidence for this trade-off, where on one end of the spectrum, we've got incredibly competitive individuals, and on the other end of the spectrum, we've got stress-tolerant individuals that could tolerate cold or dry conditions. And that was extremely exciting for us, not just because it took so long to get there, but really because it's not the first evidence of a dominance-tolerance trade-off. There's many, many papers and many systems where that's been done. But I think it's one of the cleanest examples of such a thing at such a large spatial scale. To think that there's individual fungi up in the Arctic that are structured by the same fundamental force that might be structuring individuals down in Puerto Rico was quite exciting to us. And ultimately, I mentioned we measured many of those traits. It wasn't just these niche traits or these performance traits. We actually measured many biochemical and morphological traits as well. And with over 125 traits, we decided to throw them all into a PCA. And we were delighted, in fact, to find that this dominance-tolerance trade-off came out as the best predictor of the variation across our first axis of variation. So on the one side, on the left-hand side, it's all the stress-tolerant individuals that grow, that can tolerate harsh conditions. And on the right-hand side are the competitive individuals. And now the very interesting thing that we identified with this PCA was that this axis corresponds exactly with a well-known trade-off in mycelial growth. And that is the difference between growing densely and growing quickly. If you're an organism that grows through the production of tubes, you can essentially extend very rapidly and grow your tubes outwards, or you can pile them on top of one another. But if you do that, you may be getting good protection from the environment, but you certainly can't grow as quickly as if you were just extending rapidly out. And that's exactly the trade-off we see, that the individuals that are growing very densely in a dense mat of fungus, they are able to tolerate these very harsh conditions. Whereas the individuals that are growing rapidly, they might be more metabolically active and they might be growing very quickly to dominate and tolerate space. Sorry, not tolerate space, dominate space. And I've got a lovely video that can show you that exact process. Ultimately, on the left, we've got a very slow-growing, stress-tolerant individual, and on the right is a very fast-growing, dominant individual. And when I start playing, you'll see that the fungus on the right emerges later because we added it later. But then once they hit in waves of attack, the fast-growing individual just slowly dominates its opponent. And the individual on the left starts creating all these colors and these volatile compounds and all sorts of crazy stuff that we don't even begin to understand. But what we get here is this is an example of our predictive framework. We can now predict which individuals might be likely to win in these battles and which ones will be able to tolerate in difficult environmental conditions. And that brings us back to the point of all this. Remember, we've got fungi from the tropics all the way up to the Arctic, and we just wanted to link some of these traits to those organisms. And of course, when you take any individual trait, we actually found very limited patterns, and that sort of gives us insights into why we think it seems so random. That even the drought tolerance was not well predicted by the amount of moisture and precipitation in the region, like the night sky of stars. But when we in fact combine these traits, what we end up doing is stretching out the axis. So now on one end of the spectrum, in the moist conditions, we've got dominant individuals that are not very stress tolerant. And on the other end of the spectrum, we've got tolerant individuals that are not very dominant. And by having that combination of traits, we really start to get better insights into those patterns. And it wasn't just the amount of precipitation in the area that predicted this dominance tolerance trade-off. It was in fact the amount of temperature seasonality in the area, the texture of the soil, the pH of the soil, and even the amount of organic material in the soil. And because we know we have information about each of these single variables at every single pixel at about 250 meters resolution across North America, we can once again extrapolate these patterns to every single region. Now, I should tell you, this is a map that we've done and redone 10,000 times, and it's constantly being adapted, and we're still not fully there yet. But this is the general pattern that we see, that in the harsh, dry regions and highly variable regions, these are the places that we get very stress-tolerant individuals, and in the nice blue, moist, warm places, we get these dominant individuals. And the really exciting thing about that is that this is a first predictive insight into some of these trait-based patterns, these patterns in individual level traits. And it can tell us a little bit about how those organisms function. Because the stress-tolerant individuals tend to be the individuals that are more recalcitrant. The structures of those mycelia are a lot more complicated, and we have a lot of calcium oxalate crystals and a lot of melanin and lignin that we see in those hyphae. Whereas in the blue areas, the dominant individuals are growing very fine. They are more labile in their structures. And we think that is going to translate to how efficiently they are decomposed and how efficiently they form soil organic material, although that's still very much work in process. But one thing that we can say is that this also corresponds to the rate at which they decompose wood. And Nikki Lustenhauer was the first postdoc in our lab, in fact, a couple of years ago, has just finished this analysis on measuring how these individual fungi and their traits can help us to predict decomposition rates. And she found there is some evidence within our system that the dominant individuals are faster at decomposing wood and the stress-tolerant individuals might be slightly worse. Although I would stress that in our lab study, that there's a lot of uncertainty here, and you'll notice that y-axis is log transformed, so you really have to squint to see these trends. But the very exciting thing is, she also then paired up with a great collaborator of ours, Amy Zane, who's got a study, a big field wood decomposition study in Missouri. And she has 20 different species of wood, of tree, decomposing across this large spatial gradient. And what Nikki actually found is that when we extract the fungi from those wood blocks, from those decomposing bits of wood, and we measure simply their growth rates, which, if you remember, was a reasonable predictor of that dominance tolerance trade-off. When we measure those growth rates and take a community-weighted mean value for all of those thousands of fungi, the mean growth rate of fungal growth rate in that log was one of the best predictors of wood decomposition rate, explaining about 30% of the variation in wood decomposition. That was almost as much as the variation as explained by the difference in tree species, which is obviously going to have a huge role, given that determines what compounds are being decomposed in the first place. So this is really strong, clear evidence that the attributes of a microbial community can help us to predict the turnover of carbon in a very important system. And so ultimately what we're really hoping to do is continue this line of research, building in many more species from a much, much wider environmental range, and also from a much wider phylogenetic range. So that ultimately at some point, we will be able to come back to these types of models and not just say, right, a blue pixel is dominated by this functional group and a red pixel is dominated by this functional group, but we'll even be able to approximate the variation in their trait expression within those individuals to try to see if it has a meaningful impact. And it seems like it does have a meaningful impact on the carbon turnover rate at each of those pixels across the globe. So hopefully that's going to give us better and better and better insights into what's going to happen to the amount of carbon stored on land under future climate change scenarios. So that just leaves me to thank our wonderful lab of collaborators. It's the most fantastic group. And I mentioned a lot of this was funded by the BES and Marie Curie, and our existing funders are DOB Ecology and of course ETH Zurich. So if you have any interest, please follow us at any of these social media sites.
Well, thank you ever so much for that fascinating talk, Tom. If you could perhaps stop sharing your screen so we can see you, and we'll go straight to questions, if that's all right. So the first question comes from Nadia Frontier. Apologies, Nadia, if I've mispronounced that. But this goes back to the global carbon models you were showing towards the beginning of your talk. She asked for a brief explanation of what the difference was between those global carbon models.
That's the hardest question of all because of the word brief. Essentially, so each of these models is represents thousands of mechanisms, biochemical processes that are governed by the organisms that are there, and the climate and the environment and the slope and all the features of the ecosystem. And I think a lot of the variation that we see across the models, not within the models, but across the models, is simply governed by, how people interpret ecological systems to respond, and how we think, in many models we think, okay, well the change in temperature won't have such a big difference because the organisms there will thermally acclimate, and maybe another model thinks that that's not the case, and it's those ecological mechanisms and assumptions that will lead to the huge differences in the model predictions. That was a very
great
answer.
Well, I hope that helped, Nadia. Thank you. The next question comes about your amazing battles, the fights between the fungi. When you talk about niches, are you measuring a realized niche of these fungi rather than just their fundamental niche?
Very good question. No, absolutely. None of the stuff I presented was the realized niche. This is all the fundamental niche. This is just, and it's not even a fully multi-dimensional niche. It's just an individual, what I was showing you here is the growth across an individual gradient in temperature or moisture. But what we've started to do now, and what we're really excited about, is starting to essentially do all of these battles again, but across the gradients in both temperature, moisture, and also nutrient availability. And then we're going to start, hopefully, starting to approximate at least how competitive they are across these gradients, and start to begin to build a realized niche. Although I will stress that this is all done in agar in a lab, so it's never too real.
Great, thanks. And I think I'll just try and crush this last question in as well, given the time. Do we need, in your models, as well as the fungi, to include soil fauna in the models too? How important are fauna in the soil carbon turnover?
Absolutely, I think they're incredibly important, and I certainly wouldn't want to suggest one group of organisms is more important than the other. We still don't even know that, and what we're finding is that with every new map of soil fauna or soil microbes, we can test how important they are for constraining that uncertainty. And I think that's the really exciting point that we're at with ecology, is we're beginning to get this really predictive understanding of all these different types of organisms, and start to figure out how important each level of information is for the parameter of interest. And I think when we can do that sensitivity analysis, then I'll be able to have an answer for you. But my hunch is, yes, soil organisms are incredibly important. And in fact, my PhD and early career was all based on the soil organisms feeding on the fungi to govern their activity as well. And I think they're incredibly essential.
Great. Thanks, Tom. Well, I know we've already gone over time, so thanks everyone for listening. It just remains for me to really thank Tom, thank everyone for joining us today. Next week, please join us again. We'll be hosting Holly Jones of the Northern Illinois University. She'll be talking about bison impacts on plants and animals in a world-class prairie restoration. And if you've enjoyed our talk from the British Ecological Society, why don't you consider signing up to our newsletter to find out more about all that we do, our journals, the research we publish, our events, our support for careers. If you go to our website at britishecologicalsociety.org, at the very foot of the page, there's a sign-up box, so you can find out lots more from us. And with that, I think it just remains to thank you all for listening, and have a good weekend.